Hi, I'm Dr. Hyo Kim. I'm a gastroenterologist at Gangnam Severance Hospital of Yonsei University in Korea, and one of the co-authors in the present study. We were asked by DDW to discuss a poster coming out in upcoming DDW meeting. It is titled The Effect of Multiplex Paclitaxel Emission Stent Using Pluronic Mixture Membrane in Biliary Tract. It is well known that metallic stents for malignant biliary obstruction are susceptible to occlusion by tumor growth. In order to overcome this, covered metal stents were developed to prevent tumor ingrowth by a membrane which has no anti-tumor effect. We previously reported that a metal stent covered with a Paclitaxel incorporated membranes called MIRA-1 could prevent occlusion of the stent by inhibiting tumor growth. We recently developed a new generation of Paclitaxel incorporated metal stent called MIRA-2, which was designed to deliver the drug preventing the tumor ingrowth more effectively. MIRA-2 consists of two layers. Inner layer consists of PTFE, which is resistant to biodegradation, and outer layer consists of Taxol-containing polyurethane with mixture of Pluronic F127. Pluronic F127 is a surfactant that helps releasing Paclitaxel from PU membrane. Pluronic forms the micelles, and the Paclitaxel is incorporated into the core of these micelles. This resulted in increased solubility, metabolic stability, and increased circulation time for Paclitaxel. The purpose of this study was to investigate the safety and efficacy in terms of drug delivery of a new generation Paclitaxel-eluting biliary stents. The metal stents used in the study were coated with 10% Paclitaxel, polyurethane, and one of three concentrations of Pluronic F127, which were 0, 10, and 20% respectively. Stent with each concentration was inserted in the bile duct of experimental PIC by oral transpapillary approach. After insertion of the stent, the histology of bile duct and the amount of Paclitaxel in the porcine serum were examined. Microscopically, the area where stent was inserted showed thinning and sloughing of mucosa due to compression of the stent. But as well as gross findings, there were no significant differences among four groups. The figures show microscopic findings of bile duct from 10% Pluronic group. The mucosa of central portion of the stent insert area was atrophic and showed sloughing with mild inflammation, but without fibrosis. In the proximal portion of non-stent insert area, the mucosa was preserved with intact epithelium surface and showed mild inflammation, but this change was not significantly different from other groups. In the porcine serum analysis, released Paclitaxel in 10% Pluronic F127 group was detected for 28 days. However, released Paclitaxel was detected only for 7 days in other groups. Also, to determine the efficacy of in vitro anti-tumor effect, cell viability was measured by using cell line. According to the result, 10% Pluronic F127 group showed similar cytotoxic effect of cancer cells to 20% Pluronic F127 group. In summaries of this study, first, the new Paclitaxel-eluting stent using 10% Pluronic F127 showed acceptable histologic changes in porcine bile duct. Second, the new Paclitaxel-eluting stent using 10% Pluronic F127 showed longer standing Paclitaxel release in the porcine serum than 20% Pluronic F127. Third, the new Paclitaxel-eluting stent using 10% Pluronic F127 showed cytotoxic effect similar to 20% Pluronic group. So the conclusion of our study is that the new Paclitaxel-eluting stent using 10% Pluronic F127 is relatively safe with enhanced local drug delivery and anti-tumor effect. Thank you very much for listening.